Hello there, you're welcome to Biology Video Lectures with Akerele Oladimichi Philip. On today's edition, we move on to the practical aspect of our cell biology, which talks about microscopy. Now, we'll be looking at how to use the microscope really. We've talked about some calculations, how to the part of the microscope and all of that. So today, let's look at some things here. So we want to, you see here, the aim is to use the light microscope to observe, draw, and label a section of plant and animal cell, including how to understand magnification scale. Well, we've actually done that part in other parts, some lectures before this one. So at the end of this, you should be able to know how to use light microscope to make observation and to draw specimens in biology, and you'll be able to include magnification scale. So of course, when you want to draw, the point is, a time will come where you have to look through the lens, the eyepiece of the microscope. Then we are supposed to draw what you see, not what is in textbook. What I'm saying is this: maybe you saw a worm that looks like this. From what you saw in the slide of your microscope looked like this, but the same worm was drawn like that in the textbook. You're meant to draw what you see, not what it is in textbook. For example, now you might see a bacterium cell under the slide looking somewhat like that and having maybe pili like that. Then what you have in the textbook probably was drawn like this with maybe one flagellum that way. So it's also a bacterium, but it's just different in a way. So draw what you see, not what is in the textbook. The textbook labeling is to guide you, not what you're supposed to draw really. So he says your drawing should be in proportion. Okay, you should draw what you see. I mean, it's not as if, okay, you saw something, so I mean you're supposed to draw on this paper, then you draw so tiny like that. You know, it should still become up to scale. Except you are given something to draw. I mean, you're given a scale to draw. Even when you're given the scale to draw, you won't be asked to draw so tiny like that. I mean, you're supposed to draw like, make it big to some extent. Label with straight uncrossed line. This is uncrossed. Like, for example, let's say this is the cell wall. So cell wall. This is the nucleus. Sorry, bacteria don't have nucleus. Let's say this is the this is the DNA. Now everything should be straight. Like, you literally have to put a ruler like this. Nothing like, okay, you go like this, then this is crossing. No, it shouldn't be that way. Your line should not be thick or woolly. That means that when you're drawing, you should have a simple sketch, not something very thick and woolly, like having breakages like that, or like just too thick. All of those things should be put into consideration when drawing, please. This is just a small section of that. There are more things to drawing in biology. I mean, more things. I'm going to give you a comprehensive video on that much later. So here, you're supposed to do something. For you to be able to view something on the microscope, you need to get that specimen onto something called a slide. At the beginning, I show you something. This is a slide. Yes, this is a microscope slide. That I mean, yeah, that's where you're going to put the specimen on. Of course, this is called a cover slip. We'll get to see that as we move on. But this is a slide. These are slides. These are stacks of slides. So basically here, you're supposed to put that specimen on that slide. Now, in this case here, we want to use two specimens, the chick cells and the onion cells. The chick cells, of course, comes from the human, like the normal kind of chick cell inside. Then the onion cells, like you're going to peel the onion, like the very thin layer of the onion. Now it says here that the chick cell represent the cell from animals, while the onion cell represent the cell from plants. So here, like I said at the beginning, for you to see things underneath the light microscope, it must be stained as well. So it's not enough for you to just put the thing there. So for you to view the chick cell, it must be stained with methylene blue, while the onion cells will be stained with iodine. So when you put the iodine there, that makes it visible. All right, we talked about some of this when we're doing introduction to microscopy. Now let's start with this one. Now this is, we're trying to take the onion cell now. So you're meant to cut the onion into quarters like that. Then you keep removing the layers, because these are layers like that. I believe you've seen onions before. Now as you open the layer, you're going to see this very thin membranous layer of the onions is going to be there. Now the reason why you're using that is because that's the one that the stain will be able to penetrate through, number one. Number two is that is what is easy for the light to pass through and make you see what it contains. So it's going to be very thin and transparent, so to say. So that's what you can see stopping, leaf backwards, expose the epidermis. Then number 5, epidermis is placed on the slide and covered with 2 drops of distilled water, covered with with cover slip. Now the cover slip is this thing here. It could be rectangular or spherical, as case maybe, just to hold the specimen in place, because you can't just put the specimen there like that directly. Then after you've put a drop of water, you close that, then you put a drop of the stain. In this instance, the stain is going to be iodine. Then of course the stain is drawn over the specimen using a small piece of filter paper. Just to the filter paper normally absorb things. Once you put that there, you've covered. Let me use the back of this book for example. Now this is the stage here. You already have your specimen here. Then you are putting a cover slip to cover that. Now just permit me, it's not this big really. Now the cover slip has covered it, so you can't reach your specimen easily like that anymore. So you're going to put a the filter paper here, around here. So that's going to you have soaked the filter paper in the iodine in this instance, so that will just flow through here like that's how it's going to be. So that's the reason why you're using filter paper, because you've already covered the thing already. Then you are now going to put that. So the filter paper is just to help deliver that. The other methods that can be used anyways. So that is for the onion cells. Now for the chick cell, you're going to take a cotton bud. Ideally, you should have rinsed your mouth properly before starting this experiment when it comes to the chick cell, because so that you won't bring out food debris or whatever that you have in your mouth. So it's meant to be the real chick cell. The chick cell is there all the time, because as you're chewing, brushing your teeth in the morning, some of those dead cells from the mouth are there all the time. So just take the cotton bud, you just go swab your between your lips, or sorry, your cheek, and just scrub within the cheek, or just do something like that. Then you're going to bring out something moist. That moist thing you're bringing out, of course with saliva in it, you're going to put that on your slide. Then you're going to put the cover slip as usual. Then you're going to add the stain. So in that saliva you're bringing out, it already contains the chick cell. So this is going to show us what animal cell looks like. It's going to show us what a plant cell look like. All right, so he says here, add a drop of water to the middle of the clean slide, cut up an onion and separate it out in layers like I told you. Use a tweezer. The tweezers are those things that you use to hold that. Use the same tweezer to place the epidermal tissue in the water on the slide. Add a drop of iodine solution. Place the cover slip on top of the specimen on the slide. While doing this, ensure that there are no bubbles of air in the compartment. That reminds me here, sometimes if you know how to do, you might have put the stain before putting the cover slip. Either way, you can do it that way. All right, so here so now the procedure, of course, you clip the stage. So you put the cover slip on the stage. The clip holds it there. So once the clip holds it, you put it here Everything is there. The slip, the cover slip, and the slide, the whole thing there. So this clip holds it. So this clip, stage clip, holds it. After that, the next thing is select the lowest power objective lens that is the one that has the smallest magnification on it. So maybe here you can have, maybe this is times 10, that's the smallest. So that one should be here, should be on top of the specimen. And after you're doing that, you adjust the quartz adjustment knob, which does what? Brings this whole stage up and down and brings it roughly to focus. Then the next thing you do is, of course, now you're meant to actually look through the eyepiece all the while, really. You're supposed to look through the eyepiece, select the objective lens, use the what's it called, quartz adjustment knob. Then it's going to bring it to rough focus. Then after some time, I mean, once it has brought it to rough focus, then you use your fine adjustment knob to get a clear image of what is on the slide. So if the objective lens you use is not clear enough or it's not big enough, change it. So you can change, let's say the first one you used was 10 and it was looking so small, you can change to 60 or 100 as the case may be, depending on the kind of microscope you are given. So when you change to that, you go through the same procedure again. What's the procedure? This, then this, the same way. If the specimen is standing properly, it's okay for you. If it's not, you can use this stage control to make it move either to the right or to the left, or rather to the front or to the back, like you want it to go this way or this way as the case may be. All right, so it says here, mark out the sequence of the parts you will use to view the a specimen, whatever specimen in this instance. So of course you're going to I can say this is 1. Then I'm going to select the eyepiece, sorry, the objective lens, that's 2. Of course, I mean that's after you have placed you've put the stage, you've put the slide on the stage. Then the next thing is, of course, the quartz adjustment knob, then the fine adjustment knob. So let's call this 4, let's call this 3, so to say. So it depends on I mean how you're expected to do the experiment. Of course, you should have switched on all of this. I didn't go through all the likes of switching on and all of those things. I just mean what you need to do to bring it to focus. All right. Now, so this is what the onion cell might look like or should look like underneath the microscope. So it is this blue because of the fact that it has been stained with iodine and all of that. So you can see the nucleus there. Of course, this space is cytoplasm. Then the cell membrane, this is another nucleus, this is another nucleus, cell membrane and cell wall there. So to say, it's not so distinct because it is seen by a light microscope, not electron microscope. So here again, you can see, now this is the cheek cell. These are the cheek cell. It has nucleus, cytoplasm, cell membrane. It's here. Okay, it says 400 times. It has been, so the objective lens and the eyepiece used, of course, that makes it 400 times. The onion here, this was literally drawn with like really drawn like that, so to say. So that is what this is, that simple. This is what the onion cells looks like, and also the cheek cell. All right. So this is the human cheek cell there. You can see what I just talked about. So this is it. I mean, this is someone drawing this also. You can see you're literally going to be looking at it under the microscope and drawing like that, so to say. All right, hope that was helpful.